Just to start off with, a bit of background about Genomics England. So Genomics England was set up about 2013, and the aim was to gather 100,000 full human genomes for clinical research. That included gathering the medical records as well for the people whose genomes they are. The split of the project was between rare diseases and cancer. So 50% of the genomes would come from rare diseases, and 50% of the 100,000 would come from cancer. Within cancer, there were 5 main cancer types defined, and there's 2 genomes per participant in the program because we have a germline and we have a sample of the tumour. So especially within the rare diseases, we have the trios. So we try to get the person who's affected by the rare disease and ideally the parents or siblings of that individual within the program, so as we can do sort of analysis of inheritance. With the other aim of the project was very much to lay the groundwork for introducing genetics medicine as initially a test within the NHS. So really move genome sequencing to be very similar to an MRI scan. And part of the reason for 100,000 was in some ways to force the price point down with sequencing providers. So it became affordable. And obviously the target, a full contrast MRI scan, is about £500 within the NHS. So if a whole genome sequence is going to be a viable analytic, it needs to get down to that level. But it was also to exercise the process within the National Health Service of gathering the patient data and how you actually model producing the sequences. So do you have a centralized model? Do you allow each individual hospital trust to go out and buy sequencers? There's quite a lot of sequencers already out there in the National Health Service. Are they run to full capacity? Probably not, because the reagents are incredibly expensive. So, and the time it takes for the sequencer to run through the process. So as part of this exercise, a centralised sequencing centre was set up in Cambridge, and all of the samples go to that sequencing centre, and it's scaled to be able to produce 275 sequences per day for human genomes. And that's fairly close to what we have transferred to the data centre every day from Cambridge, is to 275 sequences at about 110 gigabytes for each sequence package. So we're running at about between 7 and 8 terabytes of data transferred into the data centre per day. The other aim was to actually make the UK an attractive place to do research. If you look at economies, you have manufacturing economies and you have knowledge economies. We definitely have moved from a manufacturing economy to a knowledge economy. And there was an aim that says we have a nationalised health service so that if life sciences organisations can plug into that health service with medical records on people covering 60, 70 years, that's got to be something that's an attractive proposition to bring research to the United Kingdom. In terms of LabKey, LabKey's been used since the project started. It was really one of the very first software packages brought in to Genomics England. The work was done, the original design work and implementation work was done by Jim Davis and Ollie. The Oxford team did a lot of the work around gathering the data from the what we call genomics medicine centres. We use it for quality assurance purposes. So when a physician transfers the patient's clinical data, they have a LabKey instance that they can go to check that the data they entered as come through properly, and they can check their data down in that level, that LabKey instance. We also have a quality assurance aspect where an internal team can check the quality of the data within a LabKey instance and to carry out a medical review. So for cancer and rare disease, we have medical review teams that will ensure that the data that's coming is medically sound in correlation to the sequence samples that we receive. We've just implemented what I'd call a production tracking system. I think one of the surprises, or not surprise, it shouldn't have been a surprise, is we're running a production line for sequencing. So we collect samples at now 70 different hospitals. Those samples are then have the DNA extracted at site at that hospital. That DNA is then shipped to a central point, the biorepository in the UK, where the DNA is then QA checked and plated up onto 96 well plates and to be transferred to Illumina for them to carry out the sequencing. Knowing what you've got where in that pipeline at any time is quite important, because you can end up with sort of orphan sequences kicking around in the biorepository. You need to be able to check where we are. We need to know what our work in progress is at any time. It can take between 3 to 1 month from collection of the samples to the sample arriving at Genomics England. So we want to know what we've got arrived at the biorepository, what the biorepository have shipped to Illumina, what's failed sequencing at Illumina. And using standard LabKey, really, we're able to produce reports that when we can carry out sort of failure analysis. So we could tell, looking at it, that if you're going to get a failure in sequencing, they tend to happen as clusters on plates, on the 96 port plates. So we were looking for things such as, is there a particular row, is there a particular column on the plate that would suggest a robotics issue. We also look for low yield sequences and look back at the times taken. So, for example, esophageal cancer, unless the DNA is spun out within a certain amount of time, you're going to get a very low grade sequence down to potentially the 60-70% coverage, in which case it's still got some value but not as good. Our target is 98% coverage on all of the genomes that we receive. Other things that we've built on top of LabKey, we've just released this week a Cohort Browser. It's very much a shop window to Genomics England. Because the way our consent works, none of the sequences can leave the United Kingdom, so the researchers have to come into our environment to carry out research. So we become the IT provider to lots and lots of pharmaceutical companies and academic researchers. What you need is an environment that can be accessed that will tell you what actual disease coverages we have within there. And the other thing is, as a way of presenting the clinical research data and the index of sequences. So when a researcher comes in, they come into what we call our embassy environment, and they can get to a LabKey instance, and they can then search and build cohorts within a LabKey instance. And what that sort of looks like is we have a database. Ollie will shoot me as this is ridiculously simplistic. Postgres database called Mercury. And then the different LabKey instances look in on that Mercury database. It's segmented into different areas. So we have the major segmentation is between the research environment and our internal production environment, because within our production environment, we're dealing with patient identifiable data. And so as there's no risk of any cross-contamination, We have separate LabKey instances in separate virtual data centers where we push over the wall data sets when they're released and pseudonymized into that environment. And within the Cohort Browser, which is like the shop window, they're not just pseudonymized, they're anonymized. We're dealing with aggregated numbers of people with different medical conditions that people can look in. And this is just an example. It's very much standard LabKey. It's split between rare diseases and cancer. You can look at the overall proportions by types of diseases. So intellectual disabilities, rare blood disorders, you can see those types of things. You can drill down and look to see if we actually have coverage of a particular condition that you want to do research in, and it'll give you back the number of sequences that we have available that match those conditions. And the same with the cancer sides as well. And that we're going to start building more on, and I'll cover that. Where we're currently focusing is how can we actually give you a joined research environment. So rather than search on clinical data to look for a cohort you want to work on, and then search on the genetic data, looking at particular mutations that you're interested in. We want to have an environment where you can search on both of these environments, or in both of these types of data. And the work that we're doing as a number of strands, we are working with a number of the pharma partners and some of the academic research centres to look at what is actually the subset of data that you need to work on at the first level of research to identify your cohort. So we're doing work now looking at, can we shrink the genomic data down to a level that we can load directly into LabKey? And we've done some work with one group where we've focused on 76 genes of interest and taken a cohort of 5,000 patients, and for those genes loaded that data directly into the LabKey database, the variant data. So what you'd normally find in a variant call format file is now been loaded into LabKey as datasets. And to reduce it down even further, we've been working with some of the geneticists to say, what is the weighting on those mutations that we should apply so as we can cut out some of the noise in there? So bearing in mind the disease you're looking at, what can we say? Is it a quality threshold? Is it the number of reads that you've had to get to? So very much the initial looking at genotyping those and making that genotype data available. The next thing that we're going to be starting to work on is how we can integrate LabKey with something called OpenCGA, which is the variant database that Genomics England is using. OpenCGA is an open source project, and we are funding quite a lot of the work streams in that project. It's based at Cambridge University. We seem to have imported half of Spain to Cambridge to work on it from one university in Barcelona, who must have us on a hit list because our bioinformatics department, the primary language is Spanish. The head of our bioinformatics group is Colombian, and I think he decided he wanted to be able to speak to everyone in his department in a way that we couldn't understand. So OpenCGA ingests variant core files. So it doesn't ingest the raw read file. It ingests just the variants that are different to the norm and allows you to search across it. It's been engineered to be efficient over very large pools of data. To give you an idea of size, 100,000 variant core files will be in the region of 1.2 petabytes of storage. So at the moment, we've got it with around 20,000 sequences in. and we're close to breaking that on MongoDB, so they're redeveloping it on top of Hadoop, which is in test now, but they'll allow us to do the search. What we want to do is integrate it so as you can either access LabKey or have LabKey request the mutations within OpenCGA. I've got it working on a mini version on my laptop, so it is quite feasible. There's two ways to do it. OpenCGA has an API, but it gives you back a very large JSON file, which you're probably going to have lost the will to live by the time we've processed it in a way that LabKey can display it. So we're looking at how we can actually access the raw database behind OpenCGA from LabKey and query the data directly. I mean, it's a variant core file is actually quite simplistic in reality. They're not wildly complicated about them. They're just big. To do this work, we are working closely with academic research groups and with a number of life sciences partners. Genomics England have had a history now of about 2 years with industry, early access through something we called the Gene Consortium, where 13 pharma companies and biotechs worked with us over that period of time. We've now restructured that, and it's called the Discovery Forum. So it's not just pharma industry partners, there are other organisations with an interest have become members of that group. And what we're looking at is an iterative basis, developing new technologies based on LabKey that will allow them to actually get more access. An example of this is very much one of the companies has said, what we actually want to do is pick a gene, refine the patient base of that gene, that within that gene by people suffering from a condition, so going from 100,000 people in the program down to 200 or 300 with a particular phenotype, and then show the genotype within that disease, the breakdown. So within that disease or mutations within that disease, what are the number of people with the disease carrying that mutation? What are the number of people who don't carry that disease? And look at each of the genotypes in there. As I said, we're also working with the Wellcome Centre for Human Genetics in Oxford and working with one of their researchers to say, how can we strip the data down? What do you actually need to do research? And it's been a bit of a surprise because up until then I'd been dealing mostly with bioinformaticians. When you dealt with a geneticist, their requirements were far more simple. It was like we did an exercise where we built them one variant file for 3,000 patients for one particular gene, and we loaded it up into LabKey. We added weighting to say, I'm only interested in Mason's variants in that gene, and I'm only interested in heterozygous population. We went down from 200,000 variants in that gene to 317, and they could pick the highest signals quite easily in a graph in LabKey. And just looking at the number of variants we were finding, they could rule certain genes out as not having a relationship with the condition just because there was that much noise in there. And it was present in nearly the whole population. So out of 5,000 patients in this test, everyone had huge amounts of mutation in that gene, and there was nothing particularly common about them. So they could just draw a line under that gene as having a relationship with the condition. So that's hopefully going to mean we can move things on and actually a subset of that be put outside of the embassy environment, so as people can actually then start querying the genes and mutations within genes. So if it's a known mutation that they can find on ClinVar or EMBL or any of the other systems, they can actually see if we've got participants who have mutations in those genes, and they can decide whether we've got enough of a cohort. What we have done on the Cohort Browser which I think is going to work quite well, is we've turned on API key level access, HTTP API key level access. So a couple of the pharmaceutical companies who have LabKey instances of their own are going to integrate our LabKey environment with theirs using the API. So they won't actually log on. They'll just have their environments make HTTPS requests to LabKey from the internet to tell us. And we update that information of the available genomes once a month on the 15th of the month. And that means that they can have a milk round to see what have we released that particular month on the available genomes for research, because we have this trickle effect with genomes coming in on a daily basis from Illumina that pass through QA and then will end up in the research environment. And just to give you, this is just an example of one of the genes we were looking at. And we were looking at within the population what were the highest frequencies of those abnormalities, those mutations, and then cutting and dicing that information based on the conditions that they were looking at. So that was just an overview of what Genomics England are doing with LabKey. I mean, it really is our Swiss Army knife, is the way I describe it. It's and within the Gene Consortium, we did the wash-up session with them of what they liked about the environment, what they didn't like. And LabKey scored higher out of everything of the things that they did like about the embassy.